Hello, and welcome to our Sampled Tech Talk. My name is Harris Chowdhury. I'm the Products Marketing Manager here at Sampled, and today I'm joined by Dr. Azadeh Jadali, who is the Director of Stem Cell Lab. Azadeh, thank you for joining us. Why don't you tell us a bit about your background and what you do at Sampled?
Sure. Hi, Harris. Thank you for having me. I'm excited to be here. As you mentioned, I'm the director of the Stem Cell Lab here at Sampled. I've been here for about 2 years now. I started out as the associate director of the Stem Cell Lab, and about a year ago, I was promoted to director of Stem Cell Lab. So, in our group, we do anything and everything related to iPSCs. So, we have iPSC reprogramming services, iPSC expansion, gene editing, differentiation, specifically neurodifferentiation services. Myself, my background has always been in stem cells, so I have over 15 years of experience in the stem cell field in multiple areas in neuroscience, oncology, skin biology, 3D culture systems, and now I'm here at Sampled.
Great. Um, so it's the iPSCs that we're here to talk about today. I was wondering if you could tell us the advantages of working with iPSCs and how they differ from, say, fetal stem cells or adult stem cells.
Sure. iPSCs have a lot of advantages. In terms of comparing them to fetal stem cells, iPSCs don't have the ethical implications that fetal stem cells do. In terms of comparing them to adult stem cells, the advantage of iPSCs is that they are pluripotent, so they can differentiate into many different cell types, whereas adult stem cells tend to be multipotent at best, so you won't get the same options in terms of differentiation. Another advantage of the iPSCs is they can be patient specific or subject specific. So you can take a small tissue or blood or any type of small sample from individuals, and we can make iPSCs. So we can reprogram the cells and make iPSCs. So you basically have a better personalized medicine.
Fantastic. Can you tell us a little bit about neural progenitor cells and what the applications are for researchers who are using them?
Sure. So one advantage is with neuroprogenitor cells, you can, once you have the iPSCs, you can differentiate them into neurons and you can do various different types of studies and assays. So, for example, if you have a subject that has Alzheimer's and you want to study the neurodifferentiation and how that differs from a control or or a subject that doesn't have Alzheimer's, well, you can make neuroprogenitor cells, differentiate them into neurons, and compare to see how the neurons behave differently. And that gives you better way to do disease modeling. And then you can also use those to do drug screening assays. And also just in general, if you're doing research, doing any type of signaling pathway analysis, gene analysis, and so on and so forth to study neurodifferentiation and then potentially even regeneration.
Great. And my final question is, why would researchers outsource NPC generation to companies like Sampled?
Yeah, I mean, one of the advantages is we have the expertise to be able to do the neurodifferentiation services. Specifically, our neuroprogenitor cell differentiation is being done in partnership with Sally Temple at Neurocell. Her team has been working on the neuroprogenitor cell differentiation for quite some time, so they've perfected the process. And so clients can rest assured that they are getting the highest quality NPCs when they're outsourcing their NPC services to us. And furthermore, just in general, it's advantageous because, there's that they're going to be able to save time, resources, and they can focus on doing other aspects of their research while we're making the NPCs for them.
Great. Thank you, Azadeh, and thank you for joining us for our Sample Tech Talk. We will see you at the next one. Thank you and goodbye.